Okay, so I had put these slides up earlier at the beginning of this online transition for DNA replication and the opportunity for mutations. And this is a PowerPoint presentation I'd give to my biology class. And as part of the chemistry chapter 2 in their book and translating that on into genetics. I don't want to go through this whole thing with you. So just forgive me if I slip through some slides really fast and don't talk about it. Um, the information's here. You can look at it and read it. Mostly, I will try to point out to you what I think is important for this class and what questions I'll probably put on the test, because we've got 3 days, 4 days, 4 days. Well, by the time I get this up online detail, 3 days of the actual semester left, and I just don't want to overload you anymore. So here we go. This was somewhat already talked about with the organic molecule presentation. DNA and RNA are nucleic acids, and they're polymers of nucleotides. I am not going to ask you, which one are pyrimidines, which ones are purines, but there are 2 different types of nucleotides, different categories. The 4, you do need to know the 4 nucleotides that make up DNA, and that is thymine, cytosine, adenine, and guanine. And then there is a 5th nucleotide that replaces thymine for RNA, and that is uracil. And surely I've got that printed somewhere else, so I don't have to take time to sit here and write that out for you, but I'll keep an eye out and maybe come back to that if I don't. These nucleotides are held together by what's called a sugar phosphate backbone, and you've seen the model in the classroom, I think, and I've pointed out to you. We'll have some drawings here in a little bit. I'll try to hit that again. Okay, so here's showing the difference in bases right here. Thymine in DNA and uracil in RNA. The U standing for uracil, obviously. Excuse me, I'm frog in my throat here. The sugars, that sugar phosphate backbone, the sugar differs. In DNA, it is deoxyribose sugar, and in RNA, it is ribose sugar, and thus the name difference. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. Okay, named for the sugar and the nucleic acid. Okay, what sugar type of nucleic acid? So several different ways we can represent our molecules here. The first one here is kind of the block figure. We have this is the sugar phosphate backbone here. Okay, with the phosphate being represented by this little small, because it is small in comparison to the sugar, yellow section, and then the blue block section being the sugar group. And then you have your nucleotide coming off of it, and we have these different nucleotides for the shapes. You're going to see the bonding here a little bit, and their shape that they're given here is showing how they bond to each other. Okay, A bonds to T, you can see how that could fit in here if it's turned around, and C to G. Another way of drawing this is to give more of the molecular shape of the structures. Okay, so if you refer back to the nucleotides, in the previous example, I don't know, I actually gave you the models of those, so it's in your book, but here they are here. So here's our little phosphate group. It's labeled here, but the phosphate group, the sugar, it's a 1, 2, 3, 4, 5. It's a pentose sugar. Yeah, remember that? Here it is drawn out. Okay, so 5 carbons, but the carbons aren't actually all, whoops, what did that happen? Okay, the carbons are not actually all in the ring. We've got 1 carbon, the 5th carbon's out here as part of the bonding to the phosphate group. Okay, so that means we've got an oxygen here replacing where we'd in a carbon ring, we'd find the carbon. Okay, so then our thymine here, you're looking at this is you can see it's a 6. If there's all carbons, it'd be a 6 carbon ring structure, but we have the replacing of 2 carbons with nitrogen to make this a nucleotide. We've got some carbonyl groups here. Okay, so some complex structures that we didn't specifically talk about. The um, variations on uh, the base structures. I just kind of gave you the base structures in the organic molecules presentation. But here's where you can see. Hopefully, you can recognize some of these types of structures, and then they've got other things attached, which gives them other properties. Okay, substitutions and attachments. So, excuse me. Uh, just kind of trying to give you some examples that there's a lot more out there, obviously, than what I talked about. Just trying to give you the introductory surface ideas. Okay, so that all of that makes up the DNA. Okay, so here are the actual structures. These are pyrimidines, and these are purines. And you can see the purines are the uh, kind of a pentose ring attached to a hexose ring uh, carbon nitrogen combo structure. Okay, and the pyrimidines are single ring structures. Again, I won't ask you to draw any of these on the test or even really identify these. I don't include these in the classifications. The ones that you'll need to classify for the test are the ones that I gave in the last presentation. Okay. All right. So I'm not going to dwell anymore on this. Again, here's we use this little kind of ball model. Okay. We use this a lot of chemistry to talk about molecules. We use these different colored and size balls to show small atoms relative to larger atoms and how they all would, clump up here together in their bonds. Okay. So this is what a DNA would look like with that type of model. Uh, Watson and Crick right here are credited with finding it. I I love how they used what looks like Tinker Toys and ring stands with all sorts of clamps and attachments to represent this model. Uh, you know, uh, kind of I think it's a fun and wonderful example of kind of using what's on hand to build a model. And science, we build models. We don't understand something, we start saying, well, here's what we can tell about it. Now, what more do we need to know? And we don't know what all we need to know to start with. But okay, now here's something else. Okay, so let's add that representation in there, and on we go. Okay, so their work, however, uh, was not complete. Okay, without some information from some X-ray crystallography done by Rosalind Franklin. And of course, when they got their awards for discovering DNA, she was not credited nor mentioned anywhere along the way. Years later, it came out that her work, her contribution was pretty critical, and so she's now been posthumously uh, awarded Nobel Prize with this, and also I believe that she was, at least. Anyway, she's credited in all the citations now as being part of the research. They did not work together. You can look that up. I've read stories of controversy going on there about how actually her information was maybe possibly even stolen out of her lab. They denied ever doing it. Um, Watson's still alive to this day. He says that there was not that she didn't contribute anything to their work. As he, I haven't spoken with him personally, but that's what I've read. He has said. So still, yeah, not uh, not a settled issue. Okay, on that, but enough people outside of the situation look at the evidence and say that she contributed and deserved credit. So you'll see her name included with them now. Okay, more models and structures and some interesting things about DNA replication there that uh, is cool and I'd love to talk about, but really we're out of time. So what's important? Some of that's important. It's kind of all important, and there's so much more even than what I've got in this presentation. But that's the problem with talking about my field. This is, my field, and I could go on and on for days about this stuff. So, um, here, this is showing you where how, in a model representation with shapes, the idea that adenine and thymine bond and cytosine and guanine bond, okay, what happens is we've got a parental molecule here, and basically it separates uh, here, and then a new nucleotides are brought in and paired up, and then with that you wind up getting two new DNA copies. So this is during cell reproduction, mitosis, right before the first stage of mitosis, before uh, really before prophase and kind of what's called the interphase. This happens. Okay, this DNA synthesis happens in one of the G phases. Okay, it's called G. There's a G1, G2, and G3 phase. Okay, and so they called it interphase because they thought the cell was at rest. We couldn't see anything happening. Well, this was too small for us to see. Okay, but this is happening during that interphase. Uh, so if you've studied cell cycle in biology or zoology, I think all of you've had zoology at this point, then you should be somewhat familiar with that. Okay, and actually you probably had most of this in that. Hopefully you remember it. Anyway, this idea of what happens is the two new daughter molecules uh, the uh, have an old, but here blue is the old strand, a parental strand, and a new strand, and that's referred to as semi-conservative replication. Okay, so that's kind of a big deal because if there was an error in matching up the proper nucleotide here, the original template remains in both of these, so that there's a chance that an enzyme that comes along and proofreads can check and fix it. So it prevents mutations from happening. Uh, and if what happened was we just separated, we made a new strand in between here, and then these two grays were connected to each other and went off on their merry way. If there was an error, you wouldn't know, or you wouldn't know what the original, we don't know what the original should be. What should it have been? Which one's right? Okay, for the proofreading enzymes. So, like I said, that's a big deal in reducing the occurrence of mutations. Uh, you can read through this. This goes through the process, and like I said, it's fun and fascinating, but I don't have time to really go into this with you. The details of it. Yeah, one strand gets duplicated one way and the other one gets duplicated the other way, 5 prime, 3 prime. It's um, important in that sense that uh, replication, and it's important in my talk here about mutations. Replication can only occur uh, going from the, where's my little cursor thing here? Here we go. Going from the 3 prime toward the 5 prime direction. You see this arrow here, this gray arrow, showing that we're making this complementary strand going this way in one continuous thing. Well, the two strands, as shown here, when they pair up, they're pairing up opposite each other. Okay, the 3 prime end to this strand is here, but the 5 prime end is over here to the complementary strand. The 3 prime is over here, or whichever one you want to call template, whichever one's complementary. But they run anti-parallel is the term. So here we have a situation where this strand, this what's look like the bottom strand here, has to be replicated from the 3 prime to the 5 prime direction. So these two strands have to separate, and then there's a lag before enough of this strand here becomes separated and available for replication rather, to start. Okay, so we have a little bit done this way, and then here comes they come back in and kind of circle around, and okay, well now, oh, this is available, we can replicate this, and then, oh, hey, this is open up now, we can come up here and we can replicate that, and so we get these chunks. Okay, the lag strand is replicated in chunks, and an enzyme called the DNA ligase comes in and fuses these DNA chunks together, the segments together. This is an area that mutations can happen, mistakes can happen, replication can go wrong. Okay, and this is one of the spots where again the proofreaders come along. Okay, it is going to be attached to the template here. Okay, it doesn't get released to go on its merry way. It's going to be attached to the parental template, and so proofreaders can come along and fix those. But you've got more chance in the sense of mutation or mismatch, nucleotide mismatch happening here than you would in this area here. And so then the likelihood that a proofreader might miss something is increased as well. Okay, you got the same proofreading going on though on both strands. You've got the same mechanisms to fix anything that goes wrong there. It's just your chances of something going wrong are greater here, and therefore, the proofreading is not always perfect. Even over here, something can be missed. So the more mutations, then the greater chance something gets missed. So what DNA does for us, as I said, I think I left you on the last of the organic molecule presentation. I said DNA codes for nothing but proteins. That's what it does. The whole code of life is a code for proteins. And that I was honestly happy to some extent leaving you with that for the semester. It's like, just go think about that. That's um, strange, weird. It's got to be, there's got to be more things, it's got to be more complex than that. Well, proteins can be pretty complex. There's a lot of them, and they do a lot of things. So yeah, everything that's turned on in the body, turned off in the body. There's functional structural proteins. Um, every other structure that's not made of proteins in the cell is uh, turned on or off. The making of those and the directing of those basically is turned on and off by proteins, the chemical reactions that result in everything else that goes on, not obviously protein related, is triggered by protein. Okay, so yeah. Um, so here, what I've talked about before here is what happens for cell reproduction. We've got one cell, we need to make another. We need to make two, cell division. So that's how DNA is replicated. But the DNA code itself is information for life. And so that flow of information for all those processes in the cell, when the DNA code is actually accessed and not just copied for the next cell, uh, goes from DNA to RNA and then to a protein. And that's the process that I really want to focus in on here. And again, I'm going to skip details on it. Okay. So here's a little history of how we came about the knowledge of what goes on in different people's hypothesis. You can read that. Um, Nucleotide monomers represent letters in alphabet, and they form words in a language, basically. And basically what happens is every three bases forms what's called a triplet code. So these are little three-letter words, and we call them codons. Each word codes for one amino acid in a polypeptide or protein. So here's the DNA strand. For making a protein, we don't want to make a complementary DNA strand to our template strand. What we want to do is we want to come down here and make an RNA strand. And then from that, we're going to look at each three nucleotides, RNA nucleotides, and read that word. That codon is going to code for a specific amino acid. The next three will code for another, the next three another, and the next three another, and on down the line until you get your protein built. So that's the process I'm going to elaborate on here. Okay. All right. You can read these slides. This is the, oh, I was going to look and see if you had this in your book. I think you do. Uh, okay. This is a table. This is an RNA table, and I will show you how to use it. I usually have questions on the test where you have to take a DNA template, make an RNA strand from that, and then decode that into amino acid sequence. Okay. So I need to make sure though that you have access to this either in your book, because the test will be open book, open note, so that way you can reference this, or I'll need to make sure that uh, I may put this up as a separate document or steps, one like it, up as a separate document so you can print it off before you start the test. Okay, because you won't be able to flip out and access your notes online during the test. You need to have these printed if you want to access them. Okay. So the DNA to be transcribed is we'll have what we call the template strand, and then we're going to transcribe from that, and as I said, translate into amino acids. So all of that in words. There's this a thing about elongation and termination. Just don't worry about it. All of this process of how do we know where to start and how we know where to end in our coding. And that's all important stuff. Um, Obviously, okay, in making a protein, but due to time constraints, I really want to get to the nuts and bolts of how the mechanisms, or just how that translation and transcription happens. So we have RNA. We have we have three different types of RNA. We have mRNA, tRNA, and rRNA. The messenger RNA is the RNA we're going to be making. Okay. The transfer RNA is what is kind of attached to the amino acid that brings it to the messenger RNA strand that we've made. And the rRNA is the ribosomal RNA. It's the RNA ribosome. A ribosome is a non-membrane bound organelle. It's in the cytoplasm. It's a tiny, always designated as a tiny dot in drawings. And it is made up of an RNA and protein, two subunits, an RNA, rRNA, ribosomal RNA, and a protein. And the messenger RNA attaches at that point, and it's held there, and that is the site then of the protein synthesis, okay, of the matching of the amino acids into the proper locations to make the protein. Okay, so that's all the rRNA does. Ribosomal RNA is it's the attachment place, the point, the location of the synthesis. Um, messenger RNA brings the message of the DNA code out into the cytoplasm, and the transfer RNA transfers the amino acid units, okay, from wherever they are in the cytoplasm to that site of synthesis at the ribosome. Okay. All right. So, yeah, I'm not going to worry about breaking this down into phases and all of that for you. Okay. Um, okay. All right. That's so I just need to get me a blank. I need to get a blank screen going here and take you through it. No, I don't want to open. What am I doing? I want to file, new slide. Here we go. Lost myself. Okay. So, get a color pencil here and let's start going. All right. So my, it's called C. No, it's not called C. Undo. It's called my template DNA. So let's do a T. Okay. And then the cDNA, this would be template strand, and we'd have, if we were to make the complementary strand, it would be a cDNA, complementary DNA. Okay, so I will do that with you. I will go through that with you just once here.